and welcome to the Micro e-learning program from the Global Parkinson's Genetics Program and the Aligning Science Across Parkinson's. My name is Paula Reyes and I'm a PhD student at the Universidad Nacional Autónoma de México. In this video, I will talk about the association between MAPT H1 and H2 haplotypes with Parkinson's disease. Today, we'll be covering some key points in our presentation. We'll start by introducing what haplotypes are and why they are important to study. Then, we'll provide an overview of the MAPT gene haplotypes. Next, we'll delve into how the MAPT H1 haplotype has been associated with Parkinson's disease. And finally, we'll explore how this association might be influenced by ethnicity. Let's get started. A haplotype is a combination of alleles on a single chromosome that tend to be inherited together. They are closely located on a chromosome and rare recombination events between them. In this image, representing sequences from 6 different individuals, we observe only 3 possible combinations for the marked alleles: AGT, GTA, and AGA. These combinations are referred to as haplotypes. For a more comprehensive explanation, please check Module 2, Understanding Basic Genetic Concepts, from the PD Genetics for Non-Geneticists course on the GP2 LMS. Studying the frequency of haplotypes in case-control studies can help to identify associations between complex traits and genetic variations. Haplotype-based methods can be more effective than single nucleotide polymorphism analysis for detecting associations with diseases due to the structural and functional implications of the variants. The MAPT gene encodes the microtubule-associated protein tau and has been associated with neurodegenerative diseases, particularly tauopathies. It consists of 2 haplotypes, H1 and H2. The H1 haplotype, known as ancestral, is the most common and has been predominantly associated with increased risk for various neurodegenerative disorders, including progressive supranuclear palsy, or PSP for short, and Parkinson's disease. On the other hand, the H2 haplotype is known as inverted, as it contains an inversion of the ancestral H1 haplotype. It is less common and has been found to have a complex distribution pattern worldwide. Higher frequencies, exceeding 30%, have been observed in Southwest Asia and Southern Europe. The H1 haplotype of MAPT, specifically some subhaplotypes of H1, have been associated with the risk of Parkinson's disease. In addition to the disease risk, the H1 haplotype has been linked to specific motor phenotypes, for example, a dominant non-tremor phenotype, as well as with patterns of progression, such as cognitive decline in individuals with Parkinson's. However, inconsistent results regarding the association of H1 with Parkinson's disease across different studies involving individuals of different ancestries have also been found, suggesting that the association of the H1 haplotype could be ethnicity dependent. For instance, a study in 2006 found inconsistent results among individuals from Greece, Finland, and Taiwan. Similarly, in 2007, another study found an association of the H1 haplotype in a sample from Serbia, but not in Germany. A more recent example was published last year, analyzing the frequencies of MAPT haplotypes and their influence on the risk of Parkinson's disease by the Nigeria Parkinson's Disease Research Network. The authors did not find an association between the H1 haplotype and Parkinson's disease when adjusting for sex and age of onset. With this in mind, we can see why it's so crucial to conduct this analysis in diverse cohorts. In summary, studying haplotypes can help us reveal associations between complex traits and genetic variations. The MAPT H1 haplotype has been associated with increased risk for Parkinson's disease, as well as motor phenotypes and disease progression. However, inconsistent results across ancestries suggest that the association could be ethnicity dependent. Recent research highlights the need for cohort and diverse population analysis to improve our understanding of the impact of MAPT gene haplotypes on Parkinson's disease risk, given notable differences in the genetic architecture of this locus among diverse populations. Thank you for listening, and I hope you enjoyed this video.